Hi, everybody. Welcome to the CDCR monthly seminar. It's great to see all of you. I just have a few announcements to start. So CDCR trainees and PIs, remember that March 14th is our next CDCR trainees seminar series. And I think we're going to be hearing from Enzo and Will. So please, everyone, mark your calendars and come. And the second thing that I have been advised to mention is that the Trainee Travel Awards competition for the winter for CDCR is now open. Please submit your travel applications for all the cool conferences you want to go to. And that will be open for the next month. Okay. So as everyone is getting settled, it is my great pleasure to introduce our monthly speaker, who is, of course, from our very own institution and someone I admire a great deal, and that is Professor John Stokes. So Professor Stokes is an assistant professor with the Department of Biochemistry and Biomedical Sciences here at MAC, and he received his bachelor's in health sciences in 2011 and his PhD in 2016 in antimicrobial chemical biology, both from McMaster University. From there, he went to do his postdoctoral fellowship at the Broad Institute at MIT and Harvard, and he earned the prestigious Banting Fellowship from 2018 to 2020. Following his fellowship, we were lucky that John returned to McMaster in August of 2021 and established his research lab. And one of the Stokes Lab's primary interests is quite broadly in the application of AI approaches to develop structurally and functionally novel small molecules with defined properties. And before I sign over to John, I want to share a little analogy of how I really came to hear more about John Stokes. So McMaster University is often looking for global impact. And it's not that often that our researchers are mentioned on very huge global stages. But one morning I had turned on CNN and I was delighted to overhear that McMaster University researchers had launched the first proof of concept that AI-driven drug discovery actually works. And then that's how I got to learn about John's research. And I remember like getting off my exercise machine and cheering and going, yeah, McMaster University, global stage. So that was a really nice way to hear about Professor Stokes. And I was really happy that 2 weeks later, he contacted me to say that perhaps we should consider applying AI drug discovery to the very challenging problem of glioblastoma. So not only was I thrilled to hear about John, now I'm lucky to be working with him. And we're all very lucky that he's come to speak to the CDCR today. So John, thank you for coming.
So, hello. It must have been a slow news day. I don't know. I'm going to talk about how we use AI to find drugs. The first 2/3 is going to be antibiotics, because that's sort of like the world I live in. And then the last 1/3 is going to be cancer. And you're going to see like a huge shift in how comfortable I am in the topic. So what you should do is like pay attention for the first 2/3, and then you'll be like, all right. And then the last third is going to be a gong show. So I'm just forewarning you. Okay. Let's talk about, I should close. I should have notes here. I don't. Okay, let's talk about drugs. So I'm a microbiologist by training. So this is the problem that I spend, a good portion of my time thinking about. So this is a timeline. Does everybody know what an antibiotic is, more or less? Okay, good. All right. So we've had antibiotics now for the better part of 100 years, right? Like penicillin was accidentally discovered in 1928 by Fleming because he went on vacation and couldn't keep a clean laboratory. But we can see if we look at, the dates at which all of our current clinical antibiotics were used, we can see that they're not like equidistant as a function of time, right? We have like these hot spots of activity. So, most of our current clinical antibiotics were discovered, in the 40s to the 60s, right? This is the golden era of antibiotic discovery. And we were able to, relatively, quote, easily discover diverse structural and functional classes of antibiotics by screening secondary metabolites produced by microbes that live in dirt, right? Come the middle of the 60s, we got bad at this and we kept discovering like the same antibiotics over and over again, right? So come the middle of the 60s, 70s, into the 80s, we were like, well, if we can't discover antibiotics from the dirt anymore, we have to try, more contemporary technologies. And that's where small molecule screening came in. So, in the 90s, we started screening synthetic chemical libraries, in the hope of discovering novel structural and functional classes of antibiotics. And you can see we discovered nothing of clinical utility, right? So we're in this thing called the discovery void. What's interesting to think, though, it's like if the discovery void existed in a vacuum, we'd be okay. We still have, 20 classes of antibiotics, right? But we know that this discovery void does not exist in a vacuum, right? We have resistance to every antibiotic that's on this slide. So we see globally increasing, prevalence of antibiotic resistance, an alarmingly lean pipeline. And the question is, what do we do? So I'll put this, antimicrobial resistance problem sort of in perspective. In 2019, 1.3 million people died because of a drug-resistant infection. And if we don't discover new antibiotics soon, that number is going to become 10 million per year by 2050. And at that point, it's going to surpass cancer as a cause of death. So we need to find new antibiotics. I think that AI is a reasonable approach. I hope so, because I've bet my career on it. So we'll find out. So let's look at this, though. So on the right-hand side of the slide, we see like a conventional high-throughput screening approach, right? So what this looks like is we'll go into the lab and we'll test molecules against bacterium X, whatever we care about. These screens are generally upper bound by hundreds of thousands to maybe a couple million molecules at like the largest of scales. So we'll take these molecules, we'll screen them against our bug/target of interest, we'll get our hits, and then we'll do whatever downstream validation optimization we want. So this approach, again, this is my opinion only, and if you disagree, that's totally cool, we can talk about it, is limited for like 3 primary reasons. The first is it's very expensive, because if you want to screen 1 million molecules, you have to buy 1 million molecules. It's time consuming, because if you want to test 1 million molecules, you have to do 1 million experiments, right? And 3, we often find that, at least in the case of antibiotics, the chemical space that's explored in these chemical libraries is not ideal. Okay, so how do we overcome this? On the left side, we see what, a conventional ML-guided approach could look like. So we'll gather a training data set of compounds looking for antimicrobial activity. This could be thousands to tens of thousands. We'll then train some model. We'll talk about it. And then once we're happy with our model, we can take that model and then run predictions on in silico chemical databases, right? So it's significantly less expensive, because we only have to screen, 10,000 chemicals in the laboratory as opposed to 1 million. And once we have our trained model, we can take that model and run predictions at huge chemical spaces, hundreds of millions of molecules. And we can do that really, really quickly. So for example, where it might take, I don't know, a couple weeks to screen 10,000 molecules in the lab, we'll do 10,000 predictions in like a few minutes, right? And because we can run predictions so much more rapidly, it affords us the opportunity to explore like a truly vast chemical space in the search for molecules that do the thing that we want them to do. Is that okay? I should say, I don't mind being interrupted. And I don't care about getting through a slide deck either. So I'd much rather this be like a good learning opportunity. So like rifle a hand up if you'd like, if that's okay. Okay, perfect. All right. Yeah, slide decks don't mean anything to me. Okay. All right, cool. So one of the bugs that we're super interested in the lab is this gram-negative bacterium, Acinetobacter baumannii. It's a hospital-acquired pathogen. It's super virulent. Mortality is something like 1 in 4, something like that. And it causes a bunch of different infections like bloodstream infections, lung, meningitis, wound. And it's at the very top of the World Health Organization's list of bugs for which we need new antibiotics, as well as the CDC's list. So we were like, well, it's like this super drug-resistant, problematic pathogen. It's really, really hard to treat. Let's try to cure it, right? That's where we started. Okay. I want to talk about 2 Acinetobacter stories before we get into cancer and everything falls apart. All right, so the 2 Acinetobacter stories, we're going to talk about 2 types of modeling today at a pretty high level: discriminative modeling and generative modeling, okay? This is all in the context of discovering new chemicals to kill Acinetobacter, right? So discriminative modeling satisfies this use case. I have pictures of cats and dogs, put all the cat pictures in one pile, all the dog pictures in another, right? So this type of model takes as input pictures of cats and dogs and learns how to categorize them accordingly, right? So for the purposes of what we care about, instead of cats, replace that word with antibiotics. And instead of dogs, replace that with non-antibiotics, and you understand discriminative modeling, right? Generative modeling is a little different. Generative modeling suits this case, right? You train a model on 1 million pictures of cats. And then you say, draw me a brand new picture of a cat that doesn't look like any of the other pictures of cats you've seen, but is still a cat, right? Now replace cat with antibiotic and you're there. We good? All right, perfect. That concept took me like a year to understand. So if you got it in like 5 minutes, good for you. Okay, so we want to kill Acinetobacter using an AI model. So the first thing we have to do is gather training data, right? Here's some training data. So what we're looking at here is just growth inhibition of Acinetobacter in 384-well plates. So every dot is a different chemical. If you go low, right, where everything is red, that means we're inhibiting growth. Everything in blue does nothing, right? We know the structure of every dot up here, right? Because every dot's a chemical, and we know the structure of every chemical. So we can look at this training data set and treat this as a, quote, a binary classification test, right? Yes or no, 1 and 0. Everything in blue doesn't do anything. So we colored it blue and we labeled it a 0. Right? Everything in red is statistically antibacterial in a dish. So we colored it red and we labeled it with a 1, right? So we have structure property pairs now, and this is perfect. We can train a model to understand the structure of compounds in the context of some property, right? See, nothing fancy here. Like, this is easy stuff, right? So actually the hardest part about this is figuring out what is the best way to take the structure of a compound, which is super complicated. There's like bonds and atoms and all this stuff and whatever. Like, it's like, those are messy to look at, right? So, you know, we spend a lot of time thinking, how do we turn the structure of a chemical into a long string of numbers that can be used to train a model, right? So that long string of numbers is called a vector. So we want to take the structure of a chemical as you would draw it on a napkin and convert it to a long string of numbers that can be used for model interpretation, et cetera. That's all I'm showing here. This is, we often leverage something in the laboratory called a message passing neural network. We can get into it after. It's super nerdy. We don't need to do it now, but we'll do it after. And it's a mechanism, and it's quite a sophisticated, quite an elegant mechanism, actually, to take the structure of a compound and convert it into a long string of numbers, right? We can then take that long string of numbers, which is represented here. This is, just imagine there's a different number in every little node here. And this is the vector representation of a molecule. We can then take that vector representation and put that into something called a feed-forward neural network that takes as input, again, the vector representation, and outputs a number between 1 and 0. If that number is close to 1, it thinks the input molecule is an antibiotic. If it's close to 0, it thinks it's not an antibiotic. That's it, right? Is that good? All right, and again, we can talk about this after. But safe to say, we spend a lot of time taking structures of molecules, turning into strings of numbers, and then taking that string of numbers and through math, turning it into one number between 0 and 1. That's it. That's all that's happening. Okay, cool. I just described this. So we have our training data set, right? 7,000 whatever structure property pairs. We're going to train a neural network to understand the structure of every molecule in the context of whether it was an antibiotic or not. And then what we can do is show that trained model brand new structures it's never seen before. And then based on the atomic interconnectivity of the new input molecule, as well as what the model learned during training, it's going to output a single number between 1 and 0. If it's 1, it's an antibiotic. If it's close to 0, it's not. Good? Cool. And we can do this, you know, I say 10 million per day, not quite, close enough. Yes? Not even. It's like this string of numbers represents the structural features of an input molecule in the context of the biological system in which that molecule is placed. So this is, we can do this. It's going to get gnarly, but want to talk after, because there's actually different ways we can represent molecules. We can represent molecules as like fixed representation. So for example, see that molecule up in the top left? I can take that molecule and compute 200 like molecular features. I can then take those 200 molecular features and say, this is the vector representation for that molecule. And I can use that vector representation as my input layer for this feed-forward network. That's the most straightforward way to do it. And it works quite well, but it's not optimal. The optimally, at least in our hands, is through this message passing approach. And the distinguishing feature there is that message passing neural networks enable us to learn a different vector depending on the task at hand in order to optimize classification accuracy. So for example, I can have that same molecule being screened as an antibiotic or as an anti-cancer drug, and I will get two different vector representations because it's two different biological contexts, and those two different vector representations maximize classification accuracy depending on the task. That's a high-level description, and we can get nerdy with it later. All right, cool. All right, cool. So models. So we trained, a message passing network on those 7,500 molecules that we tested in the lab against Acinetobacter. And then the first thing we did is we ran predictions just on a tiny little chemical library that's housed at the Broad Institute, the Drug Repurposing Hub. You can see it's really small, 8,000 molecules. So we took our trained model, ran predictions on this library just to see, like, could this model work? Like, does it make sense? So what we're looking at here is prediction score of the model, right, between 0 and 1. And all we're looking at is molecule rank, right? So lowest predictions here, highest predictions here. And we can look at this and we can say, hey, most of the molecules in the Drug Repurposing Hub are not predicted to be antibiotics. And that makes sense. We know that most of the molecules in this library are not antibiotics, and that checks out. So what we care about are molecules that, exist in the upper range of prediction scores. We drew an arbitrary threshold here of a prediction score greater than 0.2. This is totally arbitrary. You can change this however you want. So that left us with, I don't know, a few hundred molecules, whatever. And then it was like, okay, well, now we don't have to look at 1,000, we can look at like 300 or something. Does that make sense? But I'll say, when we're looking for new antibiotics, we're not looking for structural analogs of stuff we already have, right? We're looking for structurally novel molecules. So out of these 300 or 400 or whatever, we said we only want to look at molecules that are structurally distinct from every known antibiotic that we've ever seen. Okay. And then we got to this, and this is a molecule that we named abicyn. And we can see this doesn't look like any existing antibiotics. I'm an antibiotic. I can't look at a cancer drug and be like, that's a cancer drug. So I don't know. Maybe you can all look at antibiotics and you're like, oh, I get it. But this doesn't look like existing antibiotics. So it has it inhibits the growth of Acinetobacter at 2 micrograms per mil, which puts it in the ballpark of existing clinical medicines for this bug. It's kind of bactericidal. So on the vertical axis here, we have viability as a function of concentration, and we kill a log-ish, which is fine. But what was super cool and what got us interested in this compound is we started testing it against clinical isolates of Acinetobacter, carbapenem-resistant Enterobacteriaceae, Pseudomonas, staph. So these are all phylogenetically diverse pathogens. And we can see that as we increase the concentration of the molecule, it can inhibit the growth of all of these Acinetobacter clinical isolates, but it doesn't do anything to Enterobacteriaceae, Pseudomonas, or staph. So we're like, huh, that's super cool. We have something that is narrow spectrum. And we did this same experiment against a whole bunch of commensals, like bugs that live in and on us that make us healthy. And we saw the same thing. So we accidentally stumbled across a narrow spectrum Acinetobacter molecule, which our model was not trained to do, by the way. This just happened to be like, a happy accident, right? It's like Bob Ross. Okay, so what we did after, it's like, well, this is an interesting observation. What is the biochemical function? So in bacterial land, often what we do when we have a new molecule and we don't know what it does and we want to figure out what it does is we just evolve resistance in the lab. And if we're lucky, the mutations will be in the target. So that's what we did here. We have wild type Acinetobacter and some with mutations with increased MICs. And when we did whole genome sequencing, all these mutations mapped in or upstream of the gene encoding this protein LOL-E. LOL-E is involved in a multi-protein complex that helps transport lipoproteins from the inner membrane of bacteria to the outer membrane. This is in gram negatives. So we got a whole bunch of mutations in LAL-E, and then we did some genetic experiments, and it turns out using CRISPR-I, if we knock down LAL-E expression, the molecule got a little bit more potent, so check, and vice versa. We have one mutant that had an intergenic mutation that was upstream of LAL-E that turned out to cause resistance because it had a basal level of expression of LAL-E that was higher than wild type, right? And then we did a wound infection on a mouse, and it worked. We did a bunch of other experiments, but I'm not going to talk about it. You can read the paper. It's good. It's a quick read too, which is what, like, we kept it short, which, like, I really like it. All right, so that's fine, cool. That's a discriminative model, right? You're all experts. You can all go do this on your own. But here's the issue with discriminative modeling. So, like I said, we can explore like millions of molecules a day using discriminative modeling. And realistically, in the real world, we're upper bound by like 1 billion. Any more than that, compute times become like way too long. You're sitting around for like months. That's not cool. And like 1 billion molecules sounds like a lot, but the theoretical totality of drug-like chemical space encapsulates something like 10 to the 60 molecules, which is like a number that you don't understand. I don't understand, you know? So like whenever I try to teach this, like our solar system has 10 to the 57 atoms in it. And that means that the total number of drug-like molecules that could theoretically exist is equivalent to the number of atoms in 1,000 solar systems. So it's like, it might as well be infinity. Like it doesn't even make sense. So we know that drug-like chemical space is huge. So the question is, how do we begin to explore more of this chemical space, you know, in a manner that doesn't take like years and years of compute time, for example? And discriminative modeling, I don't think is the way to go. I think this is where generative modeling comes in, right? Because generative modeling allows us to build compounds that are optimized for a specific task, but it's not constrained to like pre-selected in silico chemical repositories. Like it's free to explore chemical space as it sees fit, right? Does that make sense? Good. So working with a buddy of mine, James O from Stanford, we invented a new generative algorithm to design drugs. So I'm going to take a step back. Lots of people tried to play this game before. All right, like lots of generative molecular algorithms exist. The problem is, not all the time, but most, a lot of these models can build like beautiful chemicals in silico, right? And spit out like just like amazingly intricate compounds on a computer screen that you can't synthesize and test in the lab, right? Like they're synthetically intractable. And that's because, again, a lot, not all, of these generative models that have existed previously, they're not really constrained. They build molecules atom by atom. And by building molecules atom by atom, they can, you know, theoretically explore any chemical space, but hemming them in to ensure synthetic tractability is often an oversight. So we were like, if we can't build a molecule that can be synthesized and tested in the lab, we can't start. Because I'm not a computer scientist. I don't know anything about computer science, but I know what a pipette is, right? So it was like, we need to make sure that the molecules we design in the computer can be brought into the lab. How do we do that? So we figured it out. I'm going to walk, this is a paper that like got accepted like 2 weeks ago. We invented this algorithm again with my buddy James O from Stanford called SynthMol. And at a very high level, picture this, just hold this idea in your head. Instead of building molecules atom by atom, we have a stack of molecular building blocks. They're like Legos. We have 130,000 of them. And we know the reactivity and synthetic compatibility of every building block with every other building block using 13 predefined chemical reactions, right? So 130,000 building blocks, 130,000 building blocks, 13 chemical reactions, right? So we can take 2 building blocks, stick them together to build a bigger molecule. That gives us access to a chemical space of 30 billion molecules, right? So way more than we could test, like even using discriminative modeling. So I'm going to walk you through this super quick, and then everything will make sense. So we have our training data set up here in the left. We've seen this. We've already talked about this. We have a property predictor, which we've already talked about, right? But instead of going to an in silico chemical repository like the drug repurposing hub and just showing it chemicals, we have this thing, SynthMol, that inputs, right, 130,000 building blocks across 13 reactions, and it figures out how to take building blocks and stick them together in combinations that make molecules, right? Those molecules, right, here's an example of one. It's made up of 2 building blocks, is then shown to this property predictor, right? That's what we show here. And this property predictor says, hey, bud, this is a good molecule, or you missed the mark, right? If it's a good molecule, or if it's a bad molecule, that information then goes back into training SynthMol on how to search chemical space to optimally build molecules. Does that make sense? Okay, it wasn't gibberish? Okay, good. All right, cool. And then what we can do is we can go into the laboratory and synthesize them and then test them in the lab. And then we know in the real world, did we make cool molecules or did we make nothing? Right? Perfect. So training data, we have more training data for this story than the previous one, because I didn't know if this was going to work. So I was like, let's just get more training data and like cross our fingers and maybe we'll get one. All right, so we have 3 chemical libraries here. And what we're looking at here is a 2-dimensional representation of chemical space, right? In black, we have every molecule that is labeled as an antibiotic from this database called ChemBl. Then in these 3 colors, I don't know what they are because I'm colorblind. So in these 3 colors, they show the chemical space that are captured by our 3 different chemical libraries. And then with these bigger dots, which I think are red. Yeah, awesome. I got this. Those are all the hits across all 3 chemical libraries, right? So what it shows is that all of our hits are structurally super diverse. We're probably not going to hem ourselves into like a corner of chemical space. And this model, based on the totality of chemical space explored during in our training data set, should be able to generalize pretty well. That's all this is to say. Yeah. So, yeah, so this is a TISNI. We computed just like static molecular fingerprints. Yeah, so we computed 2048-bit fingerprints for every molecule, XY coordinates. Molecules that live close are similar, molecules that live far apart are, you know, of course, within like the limitations of TISNI and blah, blah, blah. Yeah. So this is just molecules that live close, live close. Okay. All right. So we actually trained 3 different property predictors using this like binarized data set, right? Blue is not a hit, red is a hit. So we trained, you know, a message passing neural network like we talked about before. We trained a message passing neural network on steroids, which we can talk about later. And then we trained a simple random forest. And they all, you know, based on in silico performance metrics, worked roughly the same. So it was a wash. All right, so this is where the actual generative part becomes interesting. So we said we want to build molecules in silico that we can build in the lab. So we needed a predefined set of molecular fragments that we knew were synthetically compatible, right? So we collaborated with folks at Enamine. Enamine's a chemical synthesis company that has something called the real space. And it's the real space that is this library that consists of, you know, 130,000 building blocks across a whole bunch of reactions, right? So we worked with them. They sent us the entire chemical file, and we are now able to build the SynthMol part of this algorithm to search this chemical space. So this is what SynthMol actually does, right? This is just the molecular generation part. All right, so let's start here. We have 130,000 building blocks. We're gonna look at all the building blocks, run all of them through that molecular property predictor, and we're gonna get ones that are strongly predicted to be antibacterial and ones that are not. What we're going to do is pick the building block that is most strongly predicted to be antibacterial on its own, okay? All right, once we have that first building block, that's level 1, we can now move to level 2. So we have our building block 1 that our model says, use this to build an antibiotic. Then what we do is say, okay, what are all the building blocks that molecule 1 is compatible with? You know, there might be 80,000. So what it does, then it looks through all 80,000 of those and says, what is the best building block to pair this with that I think is going to maximize prediction score by the predictive model, right? And it says, okay, well, it's this one, right? So we have our 2 building blocks that the model says, these are the 2 best ones to use. And then sticks them together, builds this full molecule, and then it shows that molecule to the property predictor, this full molecule. And the property predictor says, you built something great, right? So this high prediction score now is then back propagated, nerdy word. All it means is we're going to update the scores of the building blocks that went into building that molecule. And updating those scores tells this model, hey, these 2 building blocks are really good. You might want to go visit them again, right? Conversely, if this molecule was terrible and the property predictor said this is not a good molecule, that low score would be back propagated to those 2 building blocks. And it would say, don't go back here. These don't work, right? Does that make sense? Now, what's really important is you can imagine if you did this a bunch of times, you're going to go back to the same building blocks over and over and over. So we had to put in this, this function that said, don't always go back to those same building blocks, randomly explore sometimes. Right? So it's this balance between exploitation, right? Going back to stuff you know works, and exploration, stuff where you're just taking a wild stab. Right? So we do this like 20,000 times and we build molecules. Good? All right, Brad. And what's awesome is, you know, we build 20,000 molecules, lots look good, lots don't. We synthesized 58 of them. We didn't, Enamine did. There's some structural analogs here, but that's okay. There's diversity down here. And 6 of the 58 worked, so 10%. If we were to randomly select molecules from the real space and see if they were antibacterial, it'd be like, I don't know, like fractions of a percent or something. So we have 6 molecules, and then this is the actual wet lab data, and they work against Acinetobacter and staph, and they also work against other escape pathogens as well. So this was like sort of our first crack at generative modeling. And the paper is coming out hopefully in the next like few weeks in Nature Machine Intelligence, but we have a preprint up. That's like paper light though. We left a whole bunch of stuff out, but like you can go look. And I think, is the code up on GitHub?
Yes.
Awesome. Yeah. Cool. So you can go play around, or just email us if you want to play around with it. So yeah, but what's interesting and kind of annoying, it's like this paper hasn't come out yet, but we already have a better version of this that like works. So it's because the field moves too quick for publishing stuff. But yeah, if you're interested in generative like design of molecules that we can actually synthesize, like just let us know. Okay. Yeah. It's a good question. So it's largely empiric, right? So so these are tunable parameters. You can say like explore more or exploit more. If we end up if we turn exploit all the way up, we end up like basically randomly searching this chemical space, and that's reflected in the prediction scores of the molecules that are output. Most of them are terrible. Like, or conversely, if we say only exploit, we get a whole bunch of analogs of each other, like very similar molecules, and they all have really high prediction scores, but there's no diversity. So this is a case where we empirically sort of like a priori say, how much chemical diversity do we want? How many false positives are we okay with computationally? And we can adjust these parameters accordingly based on the task, right? So up front, we don't actually know. And that's where a little bit of like, you know, trial and error, like looking at molecules coming out, computing how distant are they from one another, like during generation, like how different are all these molecules, and how good or bad are the corresponding predictions. Oh yeah, but what's great, it's like we do, like when we're doing this, we can do experiments in like 20 minutes. You know, it's not like I have to do an experiment for a month before I know it doesn't work. So like, you know, when we're doing this like in silico, like model optimization type stuff, it's like, it's pretty quick. I mean, I can say it's pretty quick. They do the work. It's really quick for me, but I answer emails all day. Mhm. Mhm.
Let's say the prediction model gives something that's bad.
Mhm.
let's say. In real life, it's bad, but in the model, it is good.
Mhm.
And then it back propagate again and again.
Yeah.
How do you get that?
Yeah, so that's a perfect question. So like this approach is only as good as the property predictor itself. We know our property predictor is going to make bad guesses, and that is going to be reflected in this, Monte Carlo tree search here. So that's something like we have to live with. There's no real way around it. It's a limitation of the property predictor, which itself is a limitation of the training data to which we have access, right? So, perfect question. And the way I would lob it back to you is like, I've never done anything perfect in my life ever. I'm still pretty okay. So it's like, we're not chasing perfection necessarily. We're chasing, is this good enough for the task at hand? Is that okay? I know that's intellectually like unsatisfying. I'm sorry. I wish I had something better. Yeah. Oh yeah. Yeah, we do that. So like, remember how I said we made this better? So like we took a bunch of like these molecules and retrained the model. So like it's beginning to understand like the chemical space through which it's searching a little bit better. So yeah, but that like iterative model retraining stuff, like again, we're back in the wet lab. So it's like, it takes time. But yeah, we do, for sure we do. That part is limited, not so much by time, but like by money. You know what I mean? Cause like I'm broke. And that's not true.
How much push it and say we're not going to drive it forward and make the model go a little bit crazy, get some output molecules.
Yeah. Yeah, I mean, again, like I get cold feet, right? Like if I'm looking at a molecule that's like so crazy. So like, for example, Jeremy, who's in my lab, he's sitting right there. He like was using an AI stuff for cancer, which we're gonna get into. And he has a molecule that like legit looks like a fidget spinner. And like, I'm fully serious. And I was like, this looks interesting. And like, I want to buy it, but I can't justify it. Cause like, every fiber of my being looks at like fidget spinner molecule X. And it's like, oh boy, I know I'm going to waste $500. You know what I mean?
There's the title.
What's the title? Yeah, I know, but like, I don't know, dude. I don't know. I got a kid at home. I can't get fired right now. Yeah. So using SynthMol, the way it's currently built, we can do, what is it, 20,000 a day, 20,000 generations a day or something like that? All right, more than 20,000 a day, which is fine. It is computationally demanding, this whole tree search thing. It's memory intensive. So from like a pure like molecules per minute, if that's your argument, we can do better. But this is good enough for now. 30 billion? Oh boy, like using like a straight up like unbiased discriminative model? Oh, I don't know, like that we're probably talking like a day. But like I don't have like a supercomputer. So again, you know how it's like necessity is the mother of invention? Like I feel like that's sort of what our lab is. Like our computers are made of like bubble gum and duct tape and popsicle sticks and stuff. So yeah, if like we were like Meta, that like 30 billion would not be an issue. But we're just like John's lab. But here, I'll ruin a surprise. So Jeremy's working on a model that work totally different architecture, and it designs molecules differently. And his does like 20,000 molecules every 5 minutes or something like that. So like we're orders of magnitude faster, but it's because it's a completely different architecture and blah, 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 blah. But yeah, it's like, depending on the generative architecture, you can see like orders of magnitude swings in like number of molecules outputted per unit time. You know what I mean? All right. Let's crash and burn. All right. So, all right. All right. Can I have 10 minutes? Is that okay? All right. Okay. And if I don't make it through, it's all right. Probably better, actually. All right. So we're in antibiotics lab. And Sheila said that anecdote. I cold email her. You know why? It's because I got my first CIHR grant. I was like, I got money now. So I emailed Sheila and I was like, hey, you don't know who I am, nor do you care who I am. Can we have coffee real quick? I have an idea. And I didn't really have an idea. And then we had coffee at the corner cafe and Sheila was like the nicest person ever. And she explained to me glioblastoma, which I didn't know what it was. I don't even know what cancer is, I don't think. But she was like, this is glioblastoma. And she like, it was like the best, like for like pure like education purposes, like the best 30 minutes ever. Like I walked out of that like coffee going like, I can cure cancer now. Like, and I walked, and then, and we try to kill stuff in the lab. Like, so here's how I think about it. And it's super like elementary, but it's guided our thinking for a while and it seems to work a bit. So like, if I'm trying to cure a bacterial infection, right? I have a patient and there's all the patient cells and then bacteria in that patient that are trying to kill that patient, right? So fundamentally, all I have to do is kill the bacteria and not kill my patient and I'm good. That's it, right? And it's kind of like ditto here. If someone has cancer, it's like, I just have to kill the cancer and not kill the patient, and that's good. The problem is, right, as you get further or closer and closer to closer in like the phylogenetic tree of life, the amount of biology that overlaps increases and increases and increases, which means that the druggable targets decrease and decrease and decrease, which means the chemical space that you have to play with decreases and decreases. Does that make sense? All right. So again, AI. And I don't know, this might not work, but this is what guides a lot of our thinking. Okay, so we have very few targets to play with. We have a very small chemical space to play with amongst a giant sea of chemical spaceness, right? All right, so let's do this. Glioblastoma is brain cancer. I know that for sure. And this is why it's difficult, right? So Sheila taught me this. I didn't like, all right. So you have primary brain cancer, which is bad. And then Sheila goes and cuts it out, right? Okay, and then you cut out all the brain cancer stuff. And then you treat with temozolomide, which is actually a small molecule. It's not particularly sophisticated, but you try to treat glioblastoma with it. And then you either cut out and kill the overwhelming majority of glioblastoma cells. Then you stop therapy, and the couple cells that were still hanging out grow, and then you have recurrent GBM, which is like a super different beast than the primary one, and then you can't do it, right? When I think about this, this is the exact same as antibiotic tolerance, right? I know that because I studied antibiotic tolerance for like a while, right? I didn't say resistance. Notice how I didn't say resistance. I said tolerance. What is tolerance? If I have a population of bacteria that are isogenic, right, genetically identical, and not antibiotic resistant, there's no resistance genes here, and I take an antibiotic and I drop it in that plate, I'll kill everything except a couple, right? And those couple are just hanging out. They're metabolically inactive and they're just asleep. So they don't care if there's an antibiotic in the media because they're not doing any biology. They're taking a nap. So antibiotics break biological processes. If they're asleep and no biology is happening, they don't care. So we stop antibiotic therapy. Those cells are like, oh, this is rad. I'm going to start dividing again. And your patient's sick, right? It's ditto. It's the same problem. So it's like, okay, I'm not that smart, but I can wrap my brain around this because I've seen it before. Let's do it. The first thing that we wanted to do, so here's another TISNE, and the plots I'm going to show you are not prettied up. Like this is like no joke, like days old data that I'm going to show you. Okay. Two-dimensional representation of chemical space. Everything in blue is an antibiotic. Everything in red, here's cancer, according to Kemble. And we can look, and what I draw from this is that antibiotics and cancer drugs generally don't look the same. They occupy different regions of chemical space, and intuitively we know this, right? We can look at structures of antibiotics and know that they don't often look like cancer drugs. Okay, cool. We should be able to train a model that can now start to differentiate between all of these different chemical regions that define, for example, cancer and antibiotics. Training data, right? We've seen this. This is a screen of about 12,000 molecules. We did not do this. This was done by Basim, wherever he is. No, hey, buddy. Chitra, we didn't do this. This is way too complicated for us. And what we're looking at here is 12,000 molecules, assessing their ability to decrease viability of a patient-derived, GBM cell line in vitro. right? We're using Presto Blue as a viability indicator. If it's low, it decreased viability and we colored it red. If it is not and the cells are alive, we colored it blue, right? We've seen this before. Arbitrary hit cutoff, we said 1 standard deviation looks good. That was fine. It could have been 2 standard deviations. We could have been hyper aggressive and said it had to inhibit growth like 90%. It doesn't matter. This was fine, right? And again, we're only chasing good enough. We're not chasing perfect. So the first thing that we're doing, and this is like work in progress, is we're taking a model, a message passing neural network that's trained on this binarized data set, and we ran predictions against the Zinc 15 database, but a subset of it. So we took 12 million molecules from this virtual chemical repository. And these 12 million molecules were selected because we can go to some chemical vendor somewhere in the world and buy these molecules now, right? So this was purely a practical decision. So we ran 12 million predictions, right? Remember this curve? We've seen this before, right? So a couple molecules have high prediction scores, a bunch don't. Cool. 1 million have molecules that are above this activity threshold. So we've gone from 12 million to 1 million, which is pretty good, right? But practically, I cannot look at 1 million molecules and know which ones to buy, right? So we need subsequent filters to turn this number into a number that we can work with in the laboratory, right? All right, so we emailed SynthLab cancer experts, said, send us a small list of cancer drugs with some associated physicochemical properties, right? So we just use molecular weight and log P as two really coarse filters. So in blue, right, this distribution is every, all the molecules that were predicted here, right? And based on the cancer drugs list that they sent us, we knew the molecular weight distribution, the range that we were interested in, was no smaller than 195, no bigger than 450. For C log P, so solubility, we didn't want to go any lower than -1 or any higher than 3.5. That took us, right, from 1 million to 250-ish thousand, right? Okay, we're doing good. Let's try to do better. Still can't look at 250,000 molecules, right? Too much. All right, so then we said, I mean, brains are interesting and we got to get molecules there. So blood-brain barrier penetration is important. And so is toxicity, right? We want to kill cancer. We don't want to kill our patient, right? So we went online, found data sets that we could use to train 2 other neural networks, one that's trained to predict blood-brain barrier penetrance, one that's trained to predict the probability of a novel molecule getting clinical approval, like toxicity, like person toxicity, right? All right, so what do we want? We want a high number for blood-brain barrier permeability, right? And we want a low number for toxicity. We don't want it to be toxic, but we want it to get into brains, right? So if we look at now, yeah.
I don't know. So how did you find that model?
Yeah, so super suboptimally, but we've used this model for antibiotics and it works. So the ClinTox database is a collection of a couple thousand molecules that have gone through FDA approval and have either been denied FDA approval. And then the other column is, was it toxic? So we have molecules that are FDA approved and toxic, cancer drugs. We have molecules that are not FDA approved and not toxic, some for whatever reason, lack of efficacy, and then some that are FDA approved and not toxic. So this, we trained just on the toxicity column. And I acknowledge that lots of cancer drugs will be toxic, but given that we're looking currently at whatever, 250,000 molecules, we wanted a mechanism to get us down to the molecules that were least likely to be toxic right away. When we do this, we like for phase 1B, we're gonna rejigger this a little bit because I know like cancer drugs are toxic, but we'll figure that out when we get
But the
there.
associations are all based on the relationship the chemical has.
Yeah, yeah, 100%, yeah. Both of
It
these
has nothing
are.
to do- You don't have numbers in there that toxicity above X number of
Oh.
times per kg. It's all just, these things were called toxic. Because the chemicals look like
Yep,
them. They're
that's it.
cats.
Yeah, 100%.
Look at your dogs.
Perfect. And ditto for the blood-brain barrier
Yeah, no,
too. Yeah. Yeah. So using tox and blood-brain barrier, these two models, we went It's from 250,000 to 91,000, which is still a big number. And then what we said is, well, we want structurally novel molecules. So we said, out of all the molecules of these 91,000, if we look to see which ones are similar to hits from our training data set, there were a bunch, and that left us with 44,000. Still can't look at 44,000. So what we then did is said, here's our 44,000. We've arbitrarily picked a number. We said, cluster these into 250 structurally similar clusters. We're going to take the best predicted molecule from every cluster, right? And that's now we're at 250. And that's enough to look at, right? We can look at 250 molecules. This is just a random collection. I don't know. I don't know. Do these look like cancer drugs? Awesome. Jeremy did it. So yeah, that's, huh? Is that what they look like? There you go.
Lots of rings.
Perfect. I mean, dude. Cipro, Fluxus.
The Griston, the big ones, they're all. Anthracyclines.
That's right.
All these large 3 or 4 rings together, members.
There you go. It's all Greek to me. I don't know.
Probably are.
There you go. Jeremy, you're gonna be rich, bud. All right. All right. So this is what we're doing now. If these molecules, like we're gonna buy some, we're gonna test, I'm not gonna, Sheila's lab's gonna test them. If some of them look good, this is like perfect opportunity now to turn this into a generative modeling problem, right? We know we had some success with low-hanging fruit from molecules we could buy from the internet. But let's now get to truly novel chemical matter. And we're doing that with SynthMol, obviously, right? Because Gary developed it and it's a well-oiled machine now, right? But remember who said it? 20,000 molecules a day versus like 20,000 in 5 minutes. So we're developing a chemical language model. So this is like copy paste out of ChatGPT. I just typed in what is a chemical language model, and I did it to make a point. So ChatGPT is a thing where you type in something and it gives you something else, right? So we're building a chemical language model that is designed to output string representations of molecules and it's tuned to design molecules that satisfy the task at hand. So in us, we're trying to cure brain cancer, right? And this happens at a rate currently of 20,000 molecules every 5 minutes, and that'll get faster as we optimize. Okay, we have a lot going on. Whatever stuff. We're trying to figure out. Can we cure? Like we had an idea. All right, we're done. That's all I got. No, but nobody cares. This is just like me venting. All right. Well, we've already made SynthMol better. Chemical language models are like a couple months away from prime time. We're building like a web-based molecular property prediction tool for antibiotics. We're like one of our big tasks like that Denise is running right now. It's like we're trying to figure out how bacteria make decisions in infection sites so we better know what to target for drug development. So she's trying to figure out like how bacteria think, but like inside of infections, which is nuts. We're curing other types of bacterial infections. We're going to try to cure fungal infections, and then we're figuring out how to leverage AI-guided drug design to fix climate change through agriculture-related activities and such. I'd be careful. This is being recorded. I don't want to give away all my good ideas or mediocre ideas. All right, that's it. So this is my lab. Shouts out Jake Magdalene and James Zoe, like wicked collaborators. Sheila, of course, without saying. That's my lab. It's not the biggest lab in the world, but I'm going to say like we punch above our weight. So I'm super pumped about that. Thank you all for showing up. I'm out of time. I apologize.
Thank you, John. That was fantastic.
Was that crash and burn at the end though?
No, you did not crash and burn. You didn't go sideways at all.
You see, this is that whole
Questions for Professor Stokes? Okay, let's start with Tobias and then we'll go to Peter.
My question is how much are your models restricted by the training?
one,
How
yeah.
often do you type of fall into the same molecule? Or is these models that you generate that are they generating something truly novel that you couldn't figure out by just looking at the thing?
Yo, yeah, super good question. So this is a question about the ability of the model to generalize into novel chemical space. Okay, perfect. So Some models are better and some models are worse. So recall at the start, we talked about like static molecular representations versus these learned molecular representations, the whole message passing versus, whatever. We found that if we're looking for a model that can most reliably generalize to novel chemical space, that tends to work best using these message passing neural networks that learn like a specific vector representation of a molecule for a task, like a specific task. By design, we almost invariably look for molecules that are structurally distinct from all of our hits in our training data set. So very rarely do we even spend time looking at compounds that are analogous to what we've already seen. And so you can imagine by taking a model and forcing it to generalize into novel chemical space, there's like this tension, right? It's like we want these novel chemicals, but the model starts to make some guesses every now and then. And so here's the trade-off. As we generalize more and more and more into novel chemical space, the probability of false positives, of course, goes up, right? And we know that. So oftentimes, this is a bit of a guessing game. Like, how much can we generalize away from the chemical space on which the model was trained, not waste all of our money, but still have confidence that we will get some hits that are structurally divergent from the training data set. And we can tune our hit rate. So for example, I know if we just train a model on some chemical training data set, run predictions, and we don't filter for structural novelty, we'll get more than 50% correct, like real world predictions. But most of them are going to be analogs of what we see in training, right? So I don't have a strict answer for you. I say that the probability of false positives increases as the distance, right, in whatever n-dimensional space increases from your training data set. So we figure out a range where we're okay with some number of false positives, but we still get some that are actually active and, of course, distinct. Does that make sense?
Just a brief follow-up on that.
Yeah.
Of course, to
The thing is that most of these cancer models have been primarily designed to do something specific. For example, they want to inhibit that they want to recirculate into DNA. They want to kind of block spin because that's what they in the end do. And then are these models then finding something that's targeting something?
So perfect question. And for cancer, I don't know. But for antibiotics, I definitely know. So abicyn, the first molecule I showed you from like the Nature Chemical Biology paper, that molecule is structurally distinct from everything that the model was trained on. And it's functionally distinct from everything that it was trained on. So by having a model that could reasonably well, not perfect, but reasonably well generalize to novel chemical space, we're trying to increase the probability that those structurally novel molecules, right, they're very likely going to have novel functions, right? Because like if structure dictates function or something, I don't know, I learned that once, but like whatever. So like I'm hoping that like as we generalize into novel chemical spaces for this project, again, we have novel functions, but we don't know. We'll find out when we do the experiments.
Peter.
First of all, thank you for the awesome talk. I feel like I learned so much an hour. My question is about generative models specifically. What's to stop someone, for example, from taking one of these publicly available generative models and changing the parameter like toxicity and going from 0 to 1, and then essentially creating chemicals that can be used for very nefarious reasons? And are there any safeguards that can be put into place to kind of prevent things like that from happening?
It's a very good question. So there's nothing stopping anybody from doing whatever they want with these models because they're open source, right? So it's so funny. I'm going to flex a little bit. All right. Just a bit. So I was interviewed by someone from like McLean's or something, and they brought up this exact problem. They're like, well, how do you know that like someone's not going to try to make something terrible with this? And I said, I don't. And, are there safeguards? Probably. But if you put up a safeguard, if someone really wants to suck and do something bad, they're going to figure it out anyway. So like what I said, and I still believe this, it's like some people suck, right? But like I'm a firm believer in the fact that the number of people who want to do good vastly outnumber the number of people who suck. And like the net benefit of these technologies, I believe, is positive. But yeah, I mean, sure, we can try to invent a safeguard and it won't work, right? So I'm just like, yeah, whatever, people can suck, but like, there's more better people who, I don't know, again, super wishy-washy answer, but I'm not like a philosopher or social scientist, you know what I mean?
Well, I mean, even if you think about ChatGPT, they've recently had to put guardrails up on
Yeah.
ChatGPT, right? So probably those things hopefully will be put into place in real time. Mm.
Yeah.
Hung.
Thank you for the great talk, John. Great to see from the training data set, we learn when testing different chemicals, it's important to have their chemical space diversity. I wonder whether actually the assay and the readout diversity to see from the bacteria case and cancer case is a simple assay for growth space or proliferation space. And it's great to see it's working. But now I also see omics is on the last slide to see in the cancer, for example, there's like 1,000 genes have been monitored, which is important for cancer.
Yeah.
But also recently to consider, for example, the heterogeneity for GBM.
Mhm.
People starting to using single cell after treating with compound and to see, okay, it's killing majority of the cells, but what even survive after treating with new drugs and further developed like a resistance.
Yeah.
So with that more like a compact or diverse readout,
Mhm.
is that helpful for machine learning models or is overcome?
I mean, in a perfect world, yeah, it would be great because like you have more sophisticated like properties to pair with your molecular structures, right? Instead of just like, is there viability? Yes, no. You can have like some like high dimensional vector representation of the biological state of the system, right? So perfect. The question is like, could we gather enough training data to find like chemical, I'll say like, transcriptome pairs in order to learn a meaningful representation, right? Because like those transcriptional states, like those are super information dense, right? So and figuring out a mechanism to like, be able to draw the structure of a molecule, let's say, and predict what like the endpoint transcriptional response will be, let's say, or something. Yeah, I don't know. I don't know how much data we would need, like a bunch. I don't know. I need to think about this. Are you going to dinner? All right, cool. I'll have to think because it's a really good question, and like we've thought about it too, but like when I stopped thinking about it because like the training data that we would need is like I don't know how to get it.
Please, in the back.
Clue IO, is that what it's called? Is that cancer? Okay, if it's not antibiotics, I haven't thought about it. Okay. Oh, interesting. I should know about that. I worked at the Broad. Yeah, no, I haven't, but I don't know. I'll look into it. I don't know. Yeah, thank you for letting me know it exists. I didn't even know it existed. Yeah.
Gosh.
Just you done modeling protein combinations is building block as opposed to small molecules?
Not yet at scale. So I'll say we are working on a peptide design project that is like still a baby, but like it could have legs. If I'm so just scared to go into like the protein design space because it's getting very crowded very quick. And from a therapeutic development perspective, like I'm trying to cure whatever, like is a protein the best way to do it? I don't. So I haven't gone there. We're like dipping our toe in with antimicrobial peptides because I think we have a competitive advantage because we know bacteria really, really well. But yeah, for full-blown proteins, I have no business being here, by the way. But I have no business doing that protein designs.
Wonderful. Well, please feel free to approach John after the lecture if you have other questions, but right now help me thank him for an amazing lecture.